So basically, it's a method to analyze the links L1000 level 3 normalized gene expression dataset. So it's basically an alternative way to provide signatures for experiments, an alternative level for signatures. So here's an overview of the methods. So the methods have two parts. The first part is to identify significant experiments, which are experiments that are significantly different from controls, right? And the second is to identify significant genes, that differentially expressed genes. For the first part, I use only the 978 landmark genes. And for the second part, I use the full gene set, which is 20,000 genes. So the first part is, the result from the first part is good for experimental level analysis, which you do not want to involve any genes, such as clustering analysis. The second part is good for gene level analysis. For example, if you want to do enrichment analysis. So here's the experiment setup. I think Mario pretty much already explained it. So I want to briefly describe it here because it has the consequence on how we analyze the data. So the purple is control, the blue is the experiment. So here is the concept of batch. I think a broad view called perturbation group. So I just call it a batch. So a batch is a group of experiments that share the same plates. So here is a batch of 3 plates, and for each plate it has control replicates and the experimental replicates. I think the broad arrange the experiment as such. So for example, for one experiment, it will have the same number of replicates as the number of plates. And the replicate of the same experiment will be placed on the same value of each plate. So for example, this is the replicate in plate 1 for the experiment, and this is the second replicate, and this third replicate for the same experiment. Here's some statistics. It's about 80 control replicates per plate. And generally, there are 2 to 4 plates per batch. And so generally, each experiment will have 2 to 4 replicates. And there will be about 360 experiments per plate, because you have to subtract 384 by the number of control replicates. So here we come to the first part, identify significant experiments. Actually, the method I used is an adaptation of the character with direction method our lab published this year. This is a method invented by New Clark in our lab. I just tailored this method specifically for the links data. Here is a toy example to briefly explain this method. So now we have a gene expression measurement of 2 genes for experiment and control. Now we want to, first we use the LDA, linear discriminant analysis, to find a boundary that best segregates experiment from control in this 2 gene expression space. And then we will find the direction which point from control to experiment and perpendicular to this boundary as the direction that characterize how the experiment replicates are different from the control. So this vector will be a direction, so it will be a unit vector is a norm of 1. And this will be the single representation of the experiment. So now let's look at the projection of this direction on the axis of these two genes. So basically, this direction will be represented as a vector of two components. The first component will be the projection on gene 1, and the second projection will be the projection in gene 2. And we can see that the squared value or the absolute value of gene 1 is larger than gene 2. And so which means that the control is more different than the experiment is more different from control on gene 1 than gene 2. So the gene 1 contribute more to the difference between experiment to control, and in other words, that the gene 1 is more differentially expressed than gene 2. And then let's look at the sign of the component value, whether it's positive or negative. So the gene 1 has the positive component, which means the gene 1 is upregulating experiment, and gene 2 is, as you can see, it's negative projection. So gene 2 is downregulated in experiment. So basically, use one single direction representation. We can use the component value to describe the first, how much a gene is differentially expressed, and the second, whether the gene is upregulated or downregulated. Now we can calculate the direction for other experiment. For example, this calculate the direction for the second experiment and the direction for the third experiment. Then we can compare the similarity between experiments by use these directions. So if two directions are aligned together, which means the experiment will be more similar. If the two directions, the angle between the two extensions are large, they are not aligned to the same direction, so they are not similar. And this similarity and dissimilarity could be characterized by this Cosine distance, which is basically a measure of the angle between two directions. So I will call this to measure the similarity between experiments. This is a PC principal component analysis plot of an experiment and its replicates. So these replicates of this experiment has 4 replicates. So these are the controls for the first replicate in plate 1, and these the control replicates of the second plate. These are the control replicates of the third plate. This control of the fourth plate, and these dot are the experiments. So we can see there are systematic difference between the data measured on different plates, which certainly doesn't reflect any biology. So we call it the plate effect, and it should be corrected. So the way we correct this effect is to calculate a direction of the experiment on each plate, using the single experiment replicate and the control replicates on this plate.
Can I ask you just a very quick question? So
okay.
Is it just Q normalized observations?
Yeah. These are quantum normalized data.
Okay, but not z-scores,
so,
not Nemath.
Not the z-scores. Like we started from level 3, which are normalized data, and we derive our own signatures. Nothing related to z-score. So to correct this plate effect, so then we calculate the same number of directions as the number of replicates. So we call this characteristic direction replicates, just CD replicates. So,
Oh,
so like for you, this experiment it has 4 replicates, so it has 4 CD replicates. Then we will basically average all these CD replicates to get the direction for this experiment. Just average. Now I will introduce the concept of average cosine distance. These 3 directions are the characteristic direction replicates of one experiment. So this is the direction calculated from plate 1. This is the CD calculated from plate 2, and this CD calculated from plate 3. Remember, we use this cosine distance to measure how much 2 directions are aligned together. So we can compute the cosine distance for each 2 of these 3 CD replicates, and we get 3 distances. So the average cosine distance will be all these 3 distances averaged by 3. So this average cosine distance is basically a representation of how much these CD replicates are aligned together. So if an experiment is not significantly different from control, its CD replicates will be random. Then their CD replicates, these replicated directions will not be aligned together. And since it's a very high dimensional space, 900 or 20,000, so there's very, very little chance that that 2 replicate directions could be aligned together. So the average cosine distance will be large. But if experiment is very significantly different from control, their CD replicates should pretty much align in the same direction. So the average cosine distance will be small. So basically, small average cosine distance, the stronger the experiment, and the larger the average cosine distance, the weak the experiment. So it's basically an estimation of experimental strength. So we benchmarked this average cosine distance by evaluation of the CD method to detect the experiment strength rank. So the basic assumption is that if all the other conditions are the same, the experiment of a higher dose should elicit a stronger response. So I just correlate the dose rank, the experiment rank by dose, and the experiment strength rank for about 800 sets of experiments. And these experiments in a set are only different by dose. So here's a correlation of using average cosine distance. So it's about 0.6. And here is I use this QSS, which is a measurement of experimental strength provided in the API level 4 data sets based on these score signatures. So you can see that the average cosine distance is significantly better in the correlation between the dose rank and the experimental strength rank. Okay. But with this average cosine distance, although we can rank experiment from weak to strong, but we want to have a cutoff about to tell us at which point we will define as an experiment as significantly strong and which experiments are not significantly strong. So I use a null distribution to generate the cutoff. To calculate the null distribution, you first calculate all the CD replicates for all the experiments in a batch. So this method is based on batch, so I process batch by batch. So first I compute all the steady replicates of all the experiments in batch. So this will be the steady replicates of experiment 1, and this batch has 3 plates. The color will suggest the plate. So these are the steady replicates of experiment 1, steady replicates 2, up to steady replicates of 360. Then, from this pool of steady replicates, I random draw 3 steady replicates. For example, it might be like steady replicate of experiment 1 from plate 1, and steady replicate of experiment 9 from plate 2, and steady replicate of experiment 181 from plate 2. And then after I will treat these 3 randomly draw the steady replicates as a pseudo experiment and compute its average Cosine distance. This will be the null Cosine average Cosine distance 1. I just repeat this process by 10,000 times to get a null distribution of the average Cosine distance. Here's the histogram. Then this is basically the null distribution, and I just draw the upper 5%, which is at 0.9. So this will be the cutoff. So all the experiments which has average Cosine distance less than 0.9 will be all the significant experiments, and all the experiments that has an average Cosine distance larger than 0.9, they will not be the significant experiment. So after calculate the characteristic directions for each experiment and filter out all those experiments that are not significant, we can do some clustering analysis. Here's show some examples. So this is a multi-dimensional scaling of the character directions of the part of the links data, this RGP04 significant perturbations. And the distance between the experiment are narrowed by Cosine distance. So in this part of the links data, they are ligand perturbations to 6 different breast cancer cell lines. The ligands are classed into 4 categories: the growth factors, the interference, and this IL interference. So you can say, use this characteristic direction, ligands are separated by their class, but while you use the J-score signatures, you'll get a cluster like this, so it's pretty much overlapped. You cannot discern patterns. And actually, the character direction can reveal more subtle differences. So here, I color the same set of dots by time points instead of ligand class. So you can say, like, for some ligands, they are separated by time points. So these are the perturbations at time point 1, and these are perturbations of time point 24. But if you use this class signatures, it looks pretty much random. You can't see any patterns from this MDS plot.
Can I ask you another question, just to, we're on the same page about the Z-scores. So these Z-scores are moderated Zs?
Yeah. Like before, like Broad published the moderated Z-score for some part of the data.
Okay, thanks.
So the distance is a mirrored Euclidean distance. So you can say, in terms of clustering analysis, the the calculated direction method is pretty much better.